Hello students, how you all are? I hope you all are doing very good and you all are motivated for your boards as well as for your NEET. So beta, today the chapter we are going to start is mineral nutrition, right? So you just have to grab your NCRTs and I will request you all to learn the lecture along with me and the chapter will be done. So let's start without delaying. So the chapter is all about the mineral nutrition. That means what are nutrients? Nutrients are the compounds which are essential for any plant to grow, right? And these nutrients, they can be further of two types. That means they can be organic nutrients and they can be inorganic nutrients. Right? Organic nutrients, you know, fats, carbohydrates, proteins. These all are the organic nutrients which are used by the plant for growth and metabolism. But you know what are organic compounds, right? Which contains carbon and hydrogen together. Now, inorganic compounds, there are certain mineral elements. Which are very essential for plant to grow and to show metabolism. So, these mineral elements, they are inorganic in nature, and this chapter is all about that how these mineral elements, how these inorganic nutrients help the plant to grow, right? So, this chapter is all about mineral nutrition. So let's take it further. These are some lines of NCRT. It says that out of 105 elements discovered so far, more than 60 elements have been reported in the plants. Right? So these lines are important. Out of 105 discovered elements so far, according to NCRT, more than 60 elements are reported in plants. Now the question arises in my mind that more than 60 elements which are found present in the plants, are they all essential for the plant? Are they all good for the growth of plant? Now NCRT has taken an example. It's saying that suppose a plant is growing near a nuclear site, right? Near a nuclear site where the soil which is present there, it has a nuclear site around it. So obviously, suppose there is a radioactive, there is a radioactive element called as strontium must be there present in the soil. Because a nuclear site is in the vicinity, so the presence of radioactive material like strontium is very natural in the soil. And plant will have no choice but to take that strontium from the soil because it is present in the soil. So if I say that in some cases strontium is reported in the plants, inside the plants, so it is essential for the plants? Obviously not. It is a radioactive element. That means now I know that more than 60 elements are reported in plants, but for sure all of them are not essential for the plants, right? So now the question arises, how will I know that these 60 elements which are reported in the plants, how many of them are essential and how many of them are not essential? So I have a very good technique to know that, and the name of that technique is hydroponics. With this technique, I'll get to know which element is essential and which is not for the growth of plant. But before going to that, let's read another important line of NCRT. It says that there are some techniques like hydroponics which can detect these mineral elements at a very low concentration, and that low concentration is 10 raised to the power minus 8 grams per ml of plant matter. Remember this beta. These mineral elements which are found inside the plant, they can be detected in the plant at a very low concentration. That means it is easy. Now we have these kind of techniques which makes it easy to detect the presence of any mineral element inside the plant even at a very low concentration, and that low concentration can be 10 raised to the power minus 8 gram per ml of plant matter. This question can come directly, that at what concentration you can detect a mineral element inside the plant. It can be as low as 10 raised to the power minus 8 sorry, 10 raised to the power minus 8 gram per ml of plant matter. Okay, remember the units also. Okay, so now the question is, how will I know which element is essential and which is not? So let's get to that technique, and that is hydroponics. Hydroponics, the technique hydroponics, it was discovered by the scientist Julian von Sachs in 1860, given in NCERT. And hydroponics is also called as soil-less culture. That means we do not need soil to grow plant. We take a nutrient medium. You can see this is the diagram of NCERT. I have taken the nutrient medium in the container, right? And this nutrient medium contains everything which is essential for the growth of the plant. And what are the major things which are present in this nutrient medium? Obviously, PGRs, plant growth regulators, the plant hormones, auxin, cytokinin, right? What must be there? Obviously, energy source like sucrose, like glucose must be there, right? It should be regulated at a proper pH. It should be regulated at a proper temperature. So these things are, very regulated in this nutrient medium. And then we cover this nutrient medium and we add a aerating tube in it. What is the function of aerating tube? With the help of this aerating tube, we can add oxygen from interval to this solution, because oxygen will get consumed up, and we will stir the medium so that the medium will remain homogeneous. And this is a funnel here. With the help of this funnel, we will add nutrients to the medium after regular intervals, right? And this is the plant growing in the soil-less culture. So this is a hydroponics setup where we are growing a plant in a nutrient medium and not in soil, providing all the conditions needed for the growth of the plant. Now the question arises, how this technique will help me to know that which mineral is essential and which is not. Okay, so what we will do, suppose for instance, I want to know that magnesium is essential or not. So what will I do? I will provide every nutrient in this solution except magnesium. And then I will wait that in the absence of oxygen, this plant will be having any problem or not. So if the plant will be having some problem, like there would be yellowing on the leaves or the growth of the plant is stunted, so I will get to know, oh, magnesium is essential. Without magnesium, there are some problems which can be seen in the plant. And if, suppose for instance, I have not added strontium in it, in the solution, and I am waiting that plant will show some problem. No, the plant is growing like it's happy. It does not have any problem. That means strontium is not essential. So, whose essentiality we have to check? Just skip that mineral element in the nutrient medium and check whether if plant shows some deficiency symptom, that means that mineral element is essential. And if it doesn't show any deficiency symptom, that means the element is not essential, right? So in this way, hydroponics can be used to check the essentiality of any mineral element. And not even that, we can check even the concentration at which the mineral element is essential. Suppose per se, magnesium is essential to the plant at 0.03 ppm, right? Now I have added 0.06 ppm to the medium. So now the plant is not actually showing some good signs. That means 0.06 concentration is not good. It is needed to the plant at a particular concentration, 0.03, not below than that and not much more than that. So with the help of hydroponics, I can even know that at what particular concentration the mineral element is needed. So this technique is so helpful. So what are the applications of this technique? First of all, we can check the essentiality of any mineral element. Right? And secondly, beta, we can detect the concentration of any mineral nutrient at which it is best to the plant, of any mineral nutrient at which it is best to the plant. Right? So these are the applications of hydroponics technique. And there are some good points about hydroponics, that this kind of solution, when we grow a plant in this kind of solution, these conditions are very controlled, you know. So the plant will not be having any problem of pathogenic, contaminations. There would be no pathogenic contaminations. There will be no algal contaminations. Only the plant will grow. There will be no problem of weeding. And what is another importance of hydroponics? That in the areas where the soil is not very fertile, so we can use the hydroponics technique for those areas. And nowadays many small herbaceous plants, you cannot grow a banyan tree or a peepal tree in hydroponics, but obviously you can grow small herbaceous plants in the hydroponics, like spinach, like lettuce, like tomato, right? So these plants can be grown in soilless cultures in the controlled conditions. So these all are the importance of hydroponics, right? So if you want to add, you can add that what are the another applications or uses of hydroponics, that the plant would be, there would be no contamination. There would be no contamination, right? No algal contamination. There would be no algal contamination, and there would be no pathogenic contamination. Okay, and then you can grow many herbaceous plants with the help of this technique in the controlled conditions. Okay, so this is all about hydroponics. Now let's move on further. Now, I have just told you that with the help of the technique hydroponics, we can make it out that which element is essential and which is not. But how will you decide that a particular element is essential or not? What is the criteria for that? What is the basis for that? That yes, this mineral element will be having these properties, so it will be essential, or it will not be having these properties, so it will be non-essential. So there should be some criteria, right, on which we'll decide whether the element is essential or not, and that is called as the criteria for essentiality. Criteria for essentiality, and this criteria for essentiality was given by two scientists, Arnon and Stout. It was given by Arnon and Stout, right? So what does it say? That how will you tell that element is essential? First of all, the element should be directly involved in the growth of the plant. If that element is not given to the plant, the growth of the plant will be stunted, right? Then it should be directly involved in the process of reproduction. If that element is not given to the plant, the plant will be unable to reproduce properly. It will be unable to, produce seeds. The seed will not set in the plant. Then the plant should be irreplaceable. The mineral should be irreplaceable. That means there should be no one in the market who can replace that particular mineral element. There is no substitute for that mineral element. Only that particular mineral element can do what it can do in the plant. No one else. So there is no substitute of that particular element. And if you do not provide that mineral element to the plant, there must be some deficiency symptoms which would be shown by the plant, like yellowing of leaf or stunted growth. And that mineral element should be directly involved in the metabolism of the plant. If all these criteria are fulfilled by a particular element, then for sure it is essential mineral element. So let's read the lines of NCERT, what it is saying. The element must be absolutely necessary for normal growth and reproduction. In the absence of this element, the plant will not be able to complete its life cycle and set the seeds. It will not be able to reproduce properly. The requirement of the element must be specific, that the plant need this mineral element only, and it should not be replaceable by any other element. And the deficiency of that particular element cannot be met by supplying any other element. No, plant need that only. Okay, and the element must be directly involved in the metabolism of the plant. So these are the criteria for essentiality. Okay, now I know that out of 105 elements discovered, more than 60 are reported in the plant, but not all of them are essential. Not all those 60 elements are essential, and I can detect them in the plant at low concentration, that is 10 raised to the power minus 8 gram per ml of plant matter. With the technique of hydroponics, now I understand which are essential and which are non-essential, and how I decided them on the basis of criteria for essentiality. Now I know that out of more than 60 elements which are reported in the plant, only 17 elements, only 17 elements are essential to the plant. Okay, only 17 elements are essential to the plants. And now we will get to know about these 17 elements, right? So how many? 17. Now we will classify these 17 elements according to the quantity needed by the plant. Okay, let's move on. Now, classification of mineral elements. First, let's classify these mineral elements on the basis of quantity. On the basis of quantity needed by the plant. Okay, on the basis of the quantity, I classify all these mineral elements into two major categories, and that is macronutrients and micronutrients. Macronutrients and micronutrients on the basis of the quantity required, right? If the quantity required by the plant, if the quantity required is more than 10 millimole per kg of dry matter of plant. These words are pretty important. You know, do not confuse between them. First of all, if the quantity required by the plant is more than 10 millimole per kg of dry matter. Now what you have to remember over here, that this is millimole, right? Millimole. That means 10 raised to the power minus 3 moles. So how, in what ways it can be written? It can be written as 10 into 10 raised to the power minus 3 moles, right? This would be the same thing. Or it can be written as 0.01 moles. So this same thing can be written in various forms. So remember this beta, that this is not just it is millimole, that is 10 raised to the power minus 3 moles. Second, it is per kg, it is not gram here, it is not gram, it is kg, per kg of dry matter of plant, right? Plant has two kind of weights, one is dry weight, one is fresh weight. Fresh weight include dry weight plus the content of water in it, but dry matter is only the dry biomass, the organic biomass, excluded the amount of water. So remember, it is not the fresh weight, it is the dry matter of the plant. Clear? And if it is less than 10 millimole per kg of dry matter, then it is micronutrient. Clear? Now, these what are the role of these macronutrients? They are structural in role. That means they form the major part of the plant body. They have mainly the structural role, and they mainly they act as enzyme activators. They mainly act as enzyme activators. That means they activate the activity of enzymes. So they do not have a very peculiar structural role in plants. Okay, then the macronutrients, they are not toxic in slight excess. They are not toxic in slight excess. What does it mean? That means if suppose magnesium is required in 0.03 ppm and if I provide 0.04 ppm to the plant, it will not be toxic. But obviously if you will provide 0.08, so it will be toxic. So every mineral nutrient, if it is provided in much more quantity than it is required by the plant, every mineral element will be toxic. But yes, in slight excess, these are not toxic, but they are even toxic in slight excess. They are toxic even in slight excess. Right, then how many total essential mineral elements are there? How many? 17. So these are 9 in number and these are 8 in number. So total comes out to be 9 plus 8, 17. So these are 8 in number, these are 9 in number. You can see over here, these are the major differences between macro and micro. Now let's see which are the macronutrients. The macronutrients are carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium, sulfur, magnesium and calcium. These are the macronutrients, and the micronutrients are zinc, chlorine, copper, boron, manganese, molybdenum, iron and nickel. Right? So 1, 2, 3, 4, 5, 6, 7, 8, 9. 1, 2, 3, 4, 5, 6, 7, 8. So these are the macronutrients and these are the micronutrients. Now, classification of the mineral elements on the basis of function. So now let's discuss the classification of elements on the basis of the functions. That was on the basis of the quantity required by the plant. On the basis of functions, first of all, the elements are the structural elements, right? Obviously, all those elements which have structural role, that means the elements which forms the major plant body, they are the structural elements, and they are carbon, hydrogen, oxygen and nitrogen. Remember this. These four are the structural elements. Then comes the energy related elements or the energy related compounds. These all are given in NCERT. Energy related elements are two. One is magnesium and the another is phosphorus. Energy related, which they are related with the formation of energy, like phosphorylation reactions. In the phosphorylation reactions, ATP is formed. Phosphorus is present in the ATP. And chlorophyll, it traps the light energy and helps it in changing it into chemical energy. And inside the chlorophyll, magnesium is present. So magnesium and phosphorus, these are called as the energy related compounds or energy related elements. So this magnesium is found in chlorophyll. This is found in chlorophyll, and this phosphorus is found in ATP. Then the third category is the enzyme activators. Right? Learn them along with me here and here only. So on the basis of functions, some of the elements are called as structural elements, carbon, hydrogen, oxygen, nitrogen. Some of them are called as energy related elements, that is magnesium and phosphorus. And some of them are very good enzyme activators. That means they activate any enzyme to function properly. So a very important enzyme activator is magnesium. It's very, very important. It activates almost or major enzymes involved in respiration and photosynthesis. So it activates most of the respiratory and photosynthetic enzymes. Most of the enzymes which are involved in the process of photosynthesis and in the process of respiration, they are activated by magnesium. And some of the very good examples are, the first example which is given in NCERT is hexokinase. Hexokinase is the respiratory enzyme involved in the process of glycolysis, right? This is activated by magnesium. Then another important enzyme is Rubisco. You know that Rubisco is so important enzyme. It is involved in photosynthesis, in the dark reaction. This particular enzyme, Rubisco, is activated by magnesium, and it is a carboxylase enzyme. Right? What are carboxylases? A carboxylase enzyme is an enzyme which is responsible for the addition of CO2, and Rubisco is responsible for the addition of CO2 with RUBP. So it is a carboxylase. That means magnesium is responsible for activating the kinases and the carboxylases, because Rubisco is a kind of carboxylase. So this is important. Remember this beta, that magnesium activate carboxylases like Rubisco and kinases like hexokinase, and this element is very important for photosynthetic and respiratory enzymes. Then after this, the another important enzyme activator is zinc. Zinc activates two important enzymes. Better learn these along with me. First very important enzyme it activates is alcohol dehydrogenase. This particular enzyme is used in the alcoholic fermentation, in anaerobic respiration. And the second class of enzymes it activate are carboxylases. That means carboxylases are activated both by magnesium and zinc, and alcohol dehydrogenase is activated by zinc. Then the third very important one is molybdenum. Very important. Molybdenum is responsible for the nitrogen metabolism. It is very important for the nitrogen metabolism, and it activates two important enzymes which are involved in nitrogen metabolism. One is nitrogenase. One is nitrogenase and the another one is nitrate reductase. These are the two important enzymes which need molybdenum to work properly, and this is very important. Molybdenum very essential for nitrogen metabolism because it activates nitrogenase and nitrate reductase. Then comes manganese. Manganese is responsible for activating the complex responsible for photolysis of water. You must know this, that during photosynthesis, the water gets split up. There is splitting of water, also called as photolysis. And this photolysis is activated, this photolytic complex is majorly formed by manganese. Also by chloride ions, that I will tell you later on. And then the last one is iron. Iron activates special class of enzyme known as catalases. Catalases are responsible for the breakdown of hydrogen peroxide in the plants. So these are major enzyme activators, right? These are the structural components. These are the energy related components, manganese and phosphorus. Enzyme activator, magnesium, very important enzyme activator for respiratory and photosynthetic enzymes. Then zinc, alcohol dehydrogenase and carboxylases. Molybdenum for nitrogen metabolism, activates nitrogenase and nitrate reductase, and manganese for photolysis of water, and iron for catalases. Okay? So this is all about the mineral elements. Now we know what are mineral elements. Now we know that what are essential mineral elements, what are macro, what are micro, and what are the different roles of these, what are the different categories that these mineral elements belong to. Now let's move further. Now let's talk something about the hunger signs. What are these hunger signs, right? There is a particular concentration. There is a particular concentration of every mineral element which is required by the plant, and that concentration is called as critical concentration. If the plant gets any mineral element below the critical concentration, it shows some symptoms, some hunger signs that I want this mineral element. These hunger signs are the deficiency symptoms which are shown by the plants. So what are these hunger signs? These are the deficiency symptoms. These are the deficiency symptoms shown by plant when they get any mineral element below critical concentration. When they get any mineral element below their critical concentration, right? Below their critical concentration. And these are the following deficiency symptoms. The first one is the necrosis. Necrosis is the death of leaf tissue. The tissue which is present in the leaf, when it dies, this is called as necrosis. Chlorosis is the degeneration of chlorophyll. Chlorosis is the degeneration of chlorophyll, and because of that there is yellowing of leaf. Because of this there is yellowing of leaf. Inhibition of cell division, that means cell cannot divide properly, and delay in flowering, that the process of flowering gets delayed. So these are some important hunger signs shown by the plant. Now let's see, let's make a list that deficiency of which mineral elements cause necrosis. So there's a funda for that, there's a trick for that, and that trick is most students when we study late night, so we get hungry when we're studying up to 2 a.m. or 4 a.m. in the morning. We get hungry, and when we get hungry, there is only one instant thing which we can instantly make and eat, and that is noodles, that is Maggie. So when the student have to do these mega studies Mega studies means they have to study for a long period of time. Then they keep noodles, right? So most students keep noodles for midnight mega studies. See how I have written Z here. ZZ, mega studies. Okay, so remember this studies, ZZ. So most stands for molybdenum. Okay, most stands for molybdenum. Most students sulfur, keep potassium, noodles nitrogen, for iron, midnight manganese, mega magnesium, and studies zinc. Okay, so these all are the elements. Because of their deficiency, necrosis occurs. Will you remember the trick? Most students keep noodles for midnight mega studies. Okay, now chlorosis. There is a girl named Megha and there is a boy named Karan. Karan and Megha are in relationship, and Karan is a lot of he is not feeling good in his life. So Megha is trying to cure Karan mentally. So the question is, can Megha cure? National question. Can Megha cure Karan? So can Megha cure Karan? So it's a hot question. Can calcium, Megha magnesium, cure copper, Karan potassium? So can Megha cure Karan? Calcium, magnesium, copper, potassium. Because of their deficiency, chlorosis is seen in plants. Then inhibition of cell division is, most students keep noodles. Simple, only this much. Most students keep noodles. This much is for inhibition of cell division, and most students keep noodles for midnight mega studies is for necrosis. So obviously most stands for MO, molybdenum, sulfur, potassium, and nitrogen. And this you'll have to learn by yourself. Nitrogen, sulfur, molybdenum. Nitrogen, sulfur, molybdenum. Okay, so please beta learn these. This is very important table. Necrosis is due to the deficiency of them. Most students keep noodles for midnight mega studies. Can Megha cure Karan? Most students keep noodles, and delay. So learn them in this order, and these all are all the hunger signs shown by the plant. Okay. Now let's move on further. Yes, this is very important. This I have told you in transport in plants, that some of the elements, they are remobilized from old leaf to the younger leaves. So those elements which can move from old leaf to younger leaves, they are mobile elements. But some of the elements, they cannot move from older leaf to the younger leaves because they are structural in nature. They cannot leave that place. They will go with that leaf only. So those elements are immobile elements. So there are two kind of elements in the plant, mobile and immobile. Now, two very important statements written in NCERT are very important. Direct question comes. Elements that are actively mobilized, which are mobile in nature, their deficiency symptoms tend to appear first in the older leaves. The elements which are mobile in nature, if the plant has their deficiency, then their deficiency will be first seen in the older leaves. And the elements which are structural in nature and are not mobilized, the elements which are structural in nature, they are the inherent part of the structure, then they will not leave the place. They are not mobile, they are immobile in nature, and their deficiency first appears in the young leaves. Very important line, okay? Elements which are mobile, their deficiency first appear in the older leaf. Learn only this line, because if you learn both the statements together, you will be confused. Just learn this much. Actively mobilized elements, their deficiency first appears in older leaf, and the opposite of that in the younger leaves. Okay, now let's move further. Yes, now let's discuss about the toxicity of micronutrients. I have just told you that the micronutrients, even in slight excess, they can become toxic to the plant, even in slight excess, right? This is called as toxicity of micronutrients. Okay, now let's read some important lines of NCERT regarding this. This says that the requirement of micronutrients is always in low amounts, right? The requirement of micronutrients is very low in plants, right? While their moderate decrease cause the deficiency and a moderate increase cause toxicity. That means if they are required in 0.03 ppm, that means it should be 0.03 ppm. If they are provided at a concentration of 0.02 ppm, then the plant will show deficiency symptoms. And if it is provided at 0.04 ppm, even in slight excess, it will become toxic. That means if it is required in 0.03 ppm, that means 0.03 ppm, below which there will be deficiency symptoms, and slightly above that there will be toxicity. So what NCERT is saying, that in other words, there is a very narrow range of concentration at which the elements are optimum. That means there is a very narrow range of concentration in which we can provide these elements, these micronutrients to the plant, right? Very narrow range, 0.03 or 0.035, or right, not more than that. So there is a very narrow range of concentration in which these elements are best to the plant. Okay, any mineral ion concentration in tissues that reduces the dry weight of tissue by about 10%. This line is very important. How will you get to know that particular element is causing toxicity in the plant? One of the very important things to measure it is that if the dry weight of the plant is getting decreased by, direct question comes, if not the fresh weight, I'm repeating here, not the fresh weight, dry weight. If dry weight of the plant gets reduced by 10%, that means that plant is suffering from toxicity of micronutrient. Very important line. Then, and yes, this critical concentration at which the particular mineral micronutrient become toxic is different for different elements, right? The toxicity symptoms are difficult to identify. Again, direct question comes. Sometimes in some lines it is written, toxicity symptoms are very easy to identify. No, they are not. Because sometimes we get confused whether this particular, this particular symptom is due to toxic effect of some element, or this particular deficiency symptom or this symptom is due to deficiency of some other element. So often we get confused. So to find out the toxicity symptoms is not an easy task, right? Toxicity for any element also vary for different plants. Yes, suppose for neem plant, the toxicity level of micronutrient is different. For banyan tree, it is different. So the toxicity levels are different for different plants also. Many a times, excess of an element may inhibit the uptake of another element. And this also happens that a slight excess of one element is responsible for the reduced uptake of the other element from the soil. So these are, so interrelated that it is very tough to, actually know the toxicity symptoms. Clear? Now, toxicity of micronutrients in your syllabus, toxicity of manganese. Manganese is a micronutrient. Toxicity of manganese is discussed. So let's see what are the major Symptoms of manganese toxicity. So these are the lines of NCERT, very important. If you want to know from the outer side, if you want to know morphologically, so what are the morphological symptoms of manganese toxicity? The two prominent symptoms of manganese toxicity are, first, the brown spots, and second, the chlorotic veins. You know what are veins on the leaf? The leaf gets yellowed. There is yellowing of leaf around the area of veins. This is called as chlorotic veins. So these are the two prominent symptoms of manganese toxicity. Question comes directly. Then, what are the physiological problems that are caused due to the manganese toxicity? First, reduces the uptake of iron and magnesium. Very important. Because of the toxicity of manganese, there is reduced uptake by the plant of iron and magnesium. Prevent binding of magnesium with its enzyme. As we know that magnesium is a very good enzyme activator. The manganese does not allow, the toxicity of manganese does not allow magnesium to bind with its enzymes, and inhibits translocation of calcium to shoot apex. In the shoot apex, shoot apical meristematic cells are present which are rapidly dividing and they need calcium for division. But manganese toxicity does not allow calcium to be translocated to the shoot apex. So these are the three important things that manganese toxicity cause. Reduces the uptake of iron and magnesium, prevents binding of magnesium with its enzymes, and prevents translocation of calcium towards the shoot apex. So manganese toxicity induces combined deficiency. Very important line. Manganese toxicity induces combined deficiency of iron, magnesium and calcium. Due to slight excess in manganese, the plant has to suffer the deficiency of iron, magnesium and calcium because of these effects. Okay, so this is micronutrient toxicity, important topic again. Then let's move forward. Yes, now we have understood all the things. What are the essential elements, what are micro elements, what are different kinds of roles of elements, then what are the deficiency symptoms generally seen in case of plants, and the toxicity of micronutrients. Now let's discuss one by one what are the role of different nutrients present in the plant. Let's first go to nitrogen. Now what you have to remember here, that in what form the plant is taking that particular element from the soil. The plant takes nitrogen in the form of nitrates. The most preferable form in which the plant absorbs nitrogen from the soil is nitrate. Nitrate is, very much tasty to the plant, but it can also uptake the nitrogen in the form of nitrite and ammonium ions. This question often comes, most preferable form of nitrogen in which plant uptake it from the soil, nitrate. But it can also take it in the form of nitrite and ammonium ions. Clear? Now nitrogen is the most abundant amount of mineral element needed by the plant. Nitrogen is needed in the utmost quantity, and this is the most limiting nutrient for every kind of ecosystem, whether that is the natural ecosystem or the agricultural ecosystem, cropland of any farmer. So this is the most limiting nutrient for the growth and development of the plant. It is required in major quantities by the plant because nitrogen is present everywhere. It is present in DNA, RNA, proteins, hormones, vitamins, even in chlorophyll. Nitrogen is everywhere in the plant. That is why it was one of the structural components also. So nitrogen is required by all the plants, by the meristematic tissue, by metabolically active cells, and it is the constituent of proteins, nucleic acids, vitamins and hormones, right? And you know what happens due to deficiency of nitrogen. We have just learned necrosis, chlorosis, right? Okay, sorry, chlorosis mainly. Now, yes, now let's go to phosphorus. Phosphorus is absorbed by the plant from the soil in H2PO4 negative or HPO4 2 negative. Important. H2PO4 negative or HPO4 2 negative. Now, what are the important role of phosphorus? Phosphorus, remember, plasma membrane is bilipid in nature. So, phosphorus is the constituent of plasma membrane. Phosphorus is required in all phosphorylation reactions. What are phosphorylation reactions? You add ADP, you add phosphate to ADP, it becomes ATP. This is a phosphorylation reaction and you need phosphorus for those kind of reactions. So phosphorus is a constituent of cell membrane. Phosphorus is a constituent of cell membrane. It's a constituent of certain proteins also. Yes, it is found in nucleic acids also, DNA and RNA in the form of phosphate group. And it is found in nucleotides, yes, and is required for all phosphorylation reactions. Okay? You have learned the roles with me, I hope. Nitrogen, found everywhere. It's a structural component and the most limiting nutrient. Phosphorus, functioning of plasma membrane. It is a constituent of some proteins, nucleic acid, nucleotides, and it is important for phosphorylation reactions. Now let's move further to potassium. Potassium is taken up by the plant in the form of potassium ions, and this is very important to maintain anion-cation balance in the plant. Very important to maintain anion-cation balance in the plant. Potassium and chloride, these are the major anion-cation balancers in the plant. Second important role is it is also involved in protein synthesis. It is also involved in protein synthesis. Then it is involved in the opening and closing of stomata. Right? It was discussed in the transport in plants that during the opening of stomata, potassium ions goes into the guard cells. So they are very important in opening and closing of stomata. It is also responsible for activation of some enzymes, and it is responsible for maintaining the turgidity of cells, because when the potassium is present inside the cell, the water comes into the cell and the cells become turgid and the shape of the cell is maintained. So what are the major functions of potassium? First, it maintains anion-cation balance. Second, it is responsible for opening and closing of stomata. Third, it is responsible for the turgidity of plant cells and thus is responsible for maintaining their shape. It is also involved in protein synthesis, and it can also activate some enzymes. Clear? Then calcium. Calcium is absorbed in the form of calcium ions, and calcium is pretty important. Calcium is very important for cell division, because you must have heard of spindle fibers. They are formed during division. Spindle fibers are formed with the help of calcium ions. Calcium is very important for the formation of middle lamella. What is middle lamella? The plant cells, the two plant cells are joined together with the help of a cementing layer in between, and that cementing layer is called as middle lamella. And calcium pectate is the major component of middle lamella. Right? So let's say what NCERT has to say about it. During cell division, it is used in the synthesis of cell wall. Yes. Yes. When a plant cell divides and new cell wall has to be formed around it, calcium is very helpful in that, because the two plant cells will be joined together with the help of middle lamella. So during cell division, it is used in the synthesis of cell wall, particularly as calcium pectate in the middle lamella. So outermost part of a cell, plant cell, is middle lamella, then primary cell wall, then secondary cell wall, right? This middle lamella during division is formed by calcium as calcium pectate. It is needed during the formation of mitotic spindle. Just told you it is very important for cell division because it forms spindle fibers. It accumulates in older leaves. Yes, calcium is an immobile element. It does not move from there. It goes with the old leaf. So calcium accumulates in the older leaf because it never leaves that leaf. So calcium is an immobile element. It is involved in the normal functioning of cell membrane. It activates certain enzymes and it is important for regulating the metabolic activities, right? So what are the functions of calcium? Forms middle lamella, calcium pectate, spindle fibers, help in cell division. It helps in the normal functioning of cell membranes and regulates the metabolic activity of the cell. Okay. Now let's move on. Magnesium, absorbed by the plant from the soil in the form of magnesium ions, Mg+2. Yes, very important. I just told you that magnesium is a very important enzyme activator. It activates the major photosynthetic and respiratory enzymes like hexokinase and rubisco. I just told you there, right? It activates the enzymes of respiration and photosynthesis. Yes, we know that. And are involved in the synthesis. This is very important. A direct question comes. Which mineral element is involved in the synthesis of DNA and RNA? Magnesium. Very important question. Magnesium is a constituent of the ring structure of chlorophyll. Magnesium is present in the structure of chlorophyll, okay, and helps to maintain the ribosome structure. The two subunits of the ribosome, shorter and the larger one, they get attached due to a critical concentration of magnesium ion in the cell. At a particular concentration of magnesium ion in the cell, the structure of ribosome gets maintained. So look into my eyes and learn the functions of magnesium. First important function, very good enzyme activator. Activates respiratory and photosynthetic enzymes. Second important function, helps in the synthesis of DNA and RNA. Third important functions, maintains ribosome structure. And this magnesium is also present in the middle lamella in the form of magnesium pectate. So these are some important functions, okay? And this is present in the structure of chlorophyll. Magnesium is present in the structure of chlorophyll, right? Okay. Now sulfur. Sulfur is taken up by the plant in the form of sulfate ions. This is very important. Sulfur is present in 2 amino acids. Sulfur is present in 2 amino acids and they are cysteine and methionine. Remember this. Cysteine, methionine. Sulfur is present in them. It is the main constituent. It is the main constituent of several coenzymes. Vitamins like thiamine and biotin. Sometimes it will be written as a thymine. This will be wrong because thymine is a nitrogenous base and thiamine is a part of vitamin B complex. So it is not thymine, it is thiamine. Okay. So thiamine and biotin, though both of them are part of vitamin B complex. It is a part of coenzyme A and it is a part of very important electron carrier, that is ferredoxin. Clear? Very important for sulfur. It's a part of 2 amino acids, methionine, cysteine. It's a part of several coenzymes like coenzyme A. Then it's a part of some vitamins like thiamine and biotin and a part of ferredoxin. Clear? Now let's move on to iron. Plant take iron in the form of ferric ions. Ferric, not ferrous. Ferrous are Fe+2, not ferrous, ferric ions. Okay, remember this. Yes, in comparison to other micronutrients are required in very less quantity, but in comparison to other micronutrients, iron is required in most quantity. That means it is a micronutrient required in most quantity. Okay. It is an important constituent of proteins involved in the transfer of electrons like ferredoxin and cytochromes. You must have heard of ferredoxin in the ETS electron transport chain, right? And cytochromes are required in the electron transport chains. So both of these are the very good electron carriers in electron transport system, cytochrome and ferredoxin, and both of them have iron in them. It is reversibly oxidized. Yes, it can get converted from Fe2 plus to Fe3 plus, Fe3 plus to Fe2 plus, so releasing or accepting the electrons. So it is helpful in the redox reactions. Then it activates catalase enzyme. I just told you it activates catalase enzyme and is essential for the formation of chlorophyll. Which element is a part of chlorophyll? Magnesium. But which element is essential for the synthesis of chlorophyll? Iron. Read the question carefully. Okay. So look into my eyes and learn iron with me. First important thing, it is a part of electron carriers like ferredoxin and cytochrome. Second, it activated enzymes like catalases. It is involved in the formation of chlorophyll. It is not a part of chlorophyll. It helps in the formation of chlorophyll. And it is helpful in redox reactions. Okay. Now let's move further. Then manganese. Manganese is taken in the form of Mn2 plus ions. This is also important in which form they are taken up by the plant. It activates many enzymes involved in photosynthesis, respiration, and nitrogen metabolism. This is very important. If the question says which mineral element is essential for nitrogen metabolism, molybdenum. Molybdenum is an enzyme is a such important enzyme in the absence of which nitrogen metabolism cannot happen. So molybdenum is the must for nitrogen metabolism, but manganese can also help in nitrogen metabolism. I hope you're getting my point. So manganese is also helpful in activating many enzymes involved in photosynthesis, respiration, and nitrogen metabolism. The best defined function of manganese is the splitting of water. Very important, right? In photosynthesis, during photosynthesis, there is photolysis of water, splitting of water. Water splits and releases oxygen. So it is also called as water splitting complex or OEC, that is oxygen evolving complex. This was a question of NEET 2015. That which mineral elements form oxygen evolving complex or water splitting complex. So the answer was manganese, chloride, and traces of calcium. Very low amount of calcium, traces of calcium, they all together forms the water splitting complex. It was a question of NEET 2015. Okay, so that is why the best defined function of manganese is that it is a part of water splitting complex. Now let's go to zinc. Zn plus 2 ions. Okay, it activates various enzymes like carboxylases. I told you carboxylases, but another important enzyme it activates is alcohol dehydrogenase. I just told you. It is needed in the synthesis of auxin. This is very important. Zinc is responsible for the formation of tryptophan, which is the amino acid which is responsible for the formation of auxin. So you can say that zinc is responsible for the formation of the amino acid tryptophan, and this tryptophan is essential for the synthesis of auxin. So we say that zinc is very essential for the synthesis of auxin. Okay. Now copper. Copper is taken in the form of Cu2 plus ions. It is essential for the overall metabolism in plants, for overall metabolic activities, and it is majorly involved in redox reactions because it is changed from Cu plus to Cu plus and Cu plus to Cu plus, so helpful in redox reactions. Okay. Now comes boron, very, very important, boron. So first of all, let's learn in what form it is taken up from the soil. It is taken up BO3 3 negative, learn with me, BO3 3 negative or B4O7 2 negative, B4O7 2 negative. Now, learn boron is very important. First of all, it is required for uptake and utilization of calcium. In the absence of boron, plant will be unable to uptake calcium. That means if a plant is deficient in boron, it is by default deficient in calcium. First important function. Second, it is important for proper membrane functioning. Okay. Pollen germination, very, very important. When the pollen grain lands on stigma, pollen tube forms because of boron. If boron is not there, then pollen will not germinate and will not form pollen tube. Cell elongation. You know what is cell elongation, right? Cell differentiation, when a cell gets differentiated and performs a function. And carbohydrate translocation. You know that sucrose gets translocated through phloem and that is helped by boron. So very important examples. Okay, learn with me. First, it is important for utilization and uptake of calcium. Second, pollen germination. Third, carbohydrate translocation or sucrose translocation, that is transport of sucrose. Then cell elongation, cell differentiation and membrane functioning. Okay, then comes molybdenum. Molybdenum is taken up in the form of MoO2 2+, MoO2 2+. It is a component of several enzymes. It is a component. It is present in the enzyme nitrogenase. Molybdenum is present inside the nitrogenase enzyme and nitrate reductase, both of them which participate in nitrogen metabolism. So who is the most important mineral element for nitrogen metabolism? Molybdenum. And it is a component of nitrogenase and nitrate reductase. Chlorine, taken in the form of chloride ions. Okay, and obviously you know that it maintains anion cation balance in the cells. It forms the water splitting complex. You know it, right? So these are the major roles. Let's revise some important roles. Ready with me? Answer yourself along with me. Okay, who maintains ribosome structure? Magnesium. Which mineral element is responsible for spindle formation? Calcium. Which mineral element is responsible for DNA and RNA synthesis? Magnesium. Which element activates the most of the respiratory and photosynthetic enzymes? Magnesium. Which mineral element part of photolytic complex? Manganese chloride and traces of calcium. Which mineral element responsible for pollen germination? Boron. Which mineral element is responsible for phosphorylation reactions? Phosphorus. Which mineral element is responsible for carbohydrate translocation? Boron. Which mineral element is responsible for maintaining anion cation balance and maintaining the turgidity of plant cells? Potassium ions and chloride ions. Okay, so these are some important questions. So learn these roles, they are important beta. Now let's move forward. I have listed some important elements for you. Mobile elements are those elements which can move from older leaf to the younger leaves in the plant. And the examples are nitrogen, phosphorus, potassium, chlorine, magnesium. These all are the mobile elements. Immobile, which cannot move, they are the structural components. Calcium, sulfur, iron, boron and copper. And there is a controversy on sulfur. In the chapter Transport in Plants, sulfur is taken as mobile, and in the chapter Mineral Nutrition it is taken as immobile. So in NCERT, in chapter Transport in Plants, it is written wrong. I am telling you that sulfur is immobile and you have to learn this. Sulfur is immobile. Critical elements are those elements which are very important for plants. Plants cannot do without them, and they are nitrogen, phosphorus, potassium. Balancing elements are those elements which neutralizes the toxic effect of other elements. These are the balancing elements, and they are calcium, magnesium, and potassium. Okay, now this is the line of NCERT. Read this along with me. In addition to 17 essential elements, apart from those 17 essential elements, some beneficial elements like sodium, selenium, cobalt, silicon, they are needed by higher plants. Some higher plants, because they are complex, along with the 17 essential elements, they need some extra elements, beneficial elements for their proper functioning, and these are them: sodium, selenium, cobalt, silicon. These are not essential elements, these are beneficial elements. Okay, written in NCERT, so important. So this slide is also important. Okay, let's move further. Now, yes, this is also given in NCERT. Mechanism of absorption of minerals. This is discussed in transport in plants, but it is given in mineral nutrition, so let's discuss. So this is a root hair cell, okay? Root hair cell present in the epidermis of root. Obviously, it will be having cell wall, and below cell wall, it will be having the plasma membrane. So when the mineral elements from the soil, they will be absorbed by the root. First of all, the mineral elements will cross the cell wall, and cell wall is dead. Anything which is dead is called as apoplast, and anything which is living is called as symplast. So, first of all, the minerals will cross the cell wall. That means they will cross the dead part of the cell, that is the apoplast of the cell. And then they will cross the plasma membrane of the root hair. That means they will cross the living space of the cell. Plasma membrane is living. And then they will enter into the cytoplasm. So, when mineral elements are taken up from the soil by the root, it occurs in two phases. The first phase is called as the initiation phase. What is initiation phase? Minerals cross the dead space or the apoplast, that means the cell wall. And this phase is passive in nature. It is passive. To cross the cell wall, mineral elements, they do not require any energy because cell wall is dead. So this uptake is very rapid. This thing occurs very rapidly, fast. But the second phase is the metabolic phase, in which the minerals have to cross the living space of the cell, that is the symplast. That means they have to cross the plasma membrane, and this is active. To cross the plasma membrane, they require energy. So this uptake is a bit slower. This uptake is slow because this requires energy. Okay, so remember, whenever a root hair cell takes mineral ions from the soil, it happens in two phases: the initiation phase or the metabolic phase. In the initiation phase, the minerals crosses the cell wall or the dead space or the apoplast of the cell, that is passive and rapid. But in the metabolic phase, they cross the plasma membrane or the living space or the symplast of the cell, and this is active phase and this is slow. Okay, so let's read some lines of NCERT which are important. In the first phase, the initial rapid uptake of ions into the free space or outer space of cell, the apoplast. The first phase is the uptake of the cell into the dead space, cell wall, that is apoplast. Dead space is apoplast or cell wall, which is passive. In the second phase of uptake, the ions are taken in slowly. The ions are taken in slowly into the inner space, the symplast. The inner space of the cell, the living space of the cell, is called as symplast. The passive movement of ions into the apoplast usually occurs through ion channels, the transmembrane proteins that function as selective pores. Now, let me tell you one thing that I've just told you, that the cell wall is dead. But the cell wall also has enzymes, proteins, carrier proteins present in it. It's dead, right? It's dead, but still it acts as a first barrier. It's not that cell wall will allow everything to come. No. Like our skin also acts as a barrier, right? It protects us. Likewise, cell wall protect a plant cell. It also acts as a barrier. So yes, it's dead, but still there are certain proteins. There are certain transmembrane proteins or the ion channels present in cell wall which regulates the movement of ions through the cell wall, but that is passive in nature. That is not active. On the other hand, the entry or exit of ions to or from the symplast requires the expenditure of metabolic energy. But if anything has to cross the plasma membrane or the symplast of the cell, has to take entry into the cell or has to go out of the cell, if it involves plasma membrane or the symplast or the living space of the cell, it will require energy. Okay, clear? And the inward movement of the inward movement of the elements into the cell is called as influx, and the outward movement is called as efflux. Clear? Clear? Now let's move further. Now let's come to the important part of the chapter, and that is nitrogen cycle. If you do not, if you find something is going on fast, so you have the liberty, this is a recorded lecture, you have the liberty to pause it and to rewind it and to listen it as many times until you understand the topic. So if you do not get any point in one go, just rewind it, listen it again, just rewind it, listen it again, and make it a point that when you finish this lecture, you understand everything and you learn everything. Okay? So let's understand the nitrogen cycle. Nitrogen, I just told you that it is one of the very most important mineral elements required by the plant. It is the most, this question comes directly, which is the most limiting nutrient for both agricultural and natural ecosystems. Your answer is nitrogen. For every kind of ecosystem, nitrogen is the most limiting nutrient required by the plant. So you know that atmospheric nitrogen, what is the amount of atmospheric nitrogen? This N2 is present in atmosphere, and its concentration in the atmosphere is 78%. But in this form, in the dinitrogen form, N triple bond N, in this form, it is 78% in the atmosphere, but it is of no use of plants. Why? Because plants cannot absorb nitrogen in the form of N2. In this form, this nitrogen is of no use of plants. It is 78%, but still no use of plant, right? Because they cannot absorb it in the form of N2. So this atmospheric nitrogen is first fixed. So this process, there are four important words which are used in nitrogen cycle: nitrogen fixation, nitrification, ammonification, and denitrification. These four words together makes the nitrogen cycle, and let's understand them one by one. The very first process in the nitrogen cycle is called as fixation. What is the meaning of fixation? To convert nitrogen either in the form of ammonia or in the form of oxides of nitrogen like nitrate and nitrite, which can be used by the plant. This is fixation. What is fixation? To convert N2 into that form into that form which can be taken or can be absorbed by the plant, or that form can be ammonia, can be nitrite, or can be nitrate. That means it can be ammonia or oxides of nitrogen. Clear? So there are many types of fixation: biological nitrogen fixation, industrial nitrogen fixation, or electrical nitrogen fixation. Biological nitrogen fixation, when it is done by the bacteria. Industrial nitrogen fixation, that the industries, the automobiles, they release nitrogen in the form of oxides. Electrical nitrogen fixation, in the atmosphere, when lightning is there and the UV rays are there, in the atmosphere UV rays and lightning is there, and this UV rays and lightning have the enough energy to break these triple bonds between nitrogen and convert them into oxides of nitrogen, right? So fixation can be done with the help of industries, with the help of electrical nitrogen fixation is also done in atmosphere with the help of UV rays and lightning, and this is done by bacteria. So what we've done, we have made the soil nitrogen pool. That means the atmospheric nitrogen is fixed, is changed with the help of these processes, and it is present in the soil now. It has made the soil nitrogen pool. Then this nitrogen will be taken up by the plant. This nitrogen will be taken up by the plant, right? And the plants will, the animals will eat the plants. Then both of them, both animals and plants will die, and they will form the decaying biomass. When this dead or decaying matter will be decomposed, then ammonia will be released from it. When this dead matter will be decomposed, ammonia will release from it, and this process is called as ammonification. Right? So the ammonia will go back into the soil, and now this this process of converting ammonia into nitrite and then to nitrate, this is called as nitrification. The oxidation of ammonia to nitrite and then nitrates, this process is called as nitrification. And then the conversion of these oxides of nitrogen again to atmospheric nitrogen, when the cycle will complete, whatever has been taken must be returned, right? So this nitrites, they are again converted into N2, and this is called as denitrification. So how many processes are occurring in the nitrogen cycling? First, fixation. Second, nitrification. Third, ammonification. And fourth, denitrification. This forms the nitrogen cycle. Let's move further and discuss one by one. Let's discuss biological nitrogen fixation. That is fixation of nitrogen by microbes, by bacteria. So biological nitrogen fixation, what does it mean? Conversion of N2, conversion of nitrogen to ammonia. Conversion of nitrogen to ammonia with the help of bacteria is called as biological nitrogen fixation. Now, why? Now, many questions come, right? Why these bacteria have this capacity that they can convert N2 to ammonia? Because they have a very special enzyme, and the name of that enzyme is called as nitrogenase. Because they have this enzyme, they can convert N2 to ammonia. Okay. Now the question, a very important question. See, biological nitrogen fixation is done by only those bacteria. Okay, this capacity on earth is not given to anyone, only to bacteria. This biological nitrogen fixation capacity is given only to bacteria on earth. And that too, not to all the bacteria, only few bacteria. Only few bacteria on earth has this capacity that they can convert N2 to ammonia. And those few bacteria, they are called as diazotrophs. Those few bacteria, they are called as diazotrophs or nitrogen fixers. Right? And this is done not by all bacteria. This question comes that biological nitrogen fixation is done by all bacteria. No, only by some bacteria. And this capacity is not with the eukaryotes. So the question is, biological nitrogen fixation is done by both prokaryotes and eukaryotes? No. All prokaryotes, all bacteria? No. Some prokaryotes? Yes. All eukaryotes? No. Eukaryotes cannot fix nitrogen. Only prokaryotes, only bacteria, and that too not all bacteria, only few bacteria. And those few bacteria are called as diazotrophs. Clear? Now this table is very important. Some of the nitrogen fixers are free living in nature. That means when they are present in the soil, they alone can do this work. But some of the nitrogen fixers, they need help of some other plant to do this thing. So they are symbiotic nitrogen fixers. Okay. So free living fixers are independently fix nitrogen in the soil. They can do it independently in the soil. And they can be aerobic in nature and they can be anaerobic in nature. This question comes directly, pretty important. These are some examples given here. More than enough for NEET. More than enough for NEET, and direct question comes. Beijeringia, Azotobacter, Azospirillum, all these three are aerobic bacteria which are free living nitrogen fixers. Klebsiella, Clostridium, Bacillus, Rhodospirillum, Rhodopseudomonas, these all are anaerobic, free living nitrogen fixers. These three are heterotrophs in nature. These two are photosynthetic bacteria, but still they can fix nitrogen also. So they are nitrogen fixers also. Clear? If you learn these examples, more than enough for NEET. Then some of the bacteria, they can fix nitrogen when they are in symbiotic association with other plant. One of the very important example is Rhizobium. Rhizobium forms symbiotic association with leguminous plants. And they are present inside their root nodules. Second very important example, which is very important because it is given in two places in NCERT, Frankia. Frankia is a symbiotic nitrogen fixer which forms symbiotic association with non-leguminous plants like Alanus, Casuarina, Myrica. These are the three plants with which Frankia is found associated. So this table is pretty important. Have a look on it. It's pretty important table. All the examples given over here are enough for NEET. Okay. So can we move forward now? Now, blue-green algae. You must have heard the name of cyanobacteria, the blue-green algae. They are also very important nitrogen fixers. These are the example of the cyanobacteria which can do this in free living condition. Olocera, Tolipothrix. This question has come in NEET. Olocera is a free living cyanobacteria which can fix nitrogen, and Tolipothrix, and they are found in rice or paddy fields. They are not found in symbiotic association with rice. No. They are found in rice field, but they fix nitrogen in free living condition. Trichodesmium is a red algae which, oh my god, what I'm saying. It's a blue-green algae which cause red sea. So it is also a nitrogen fixer. nostoc, anabina, these all are free living nitrogen fixing cyanobacteria. Then, very important, nostoc and anabina are special because they can fix nitrogen both in free living condition as well as in symbiotic condition. Nostoc is found in symbiotic association with a bryophyte, that is Anthoceros, and anabina is found in symbiotic association with Cycas, a gymnosperm, and Azolla, an aquatic pteridophyte. Right? So these are the important blue-green algae which are nitrogen fixers. These are free living and these are symbiotic. Now nitrification. What is the meaning of nitrification? Oxidation of ammonia. Oxidation of ammonia to nitrite or nitrate. And this process, the process which is done by these bacteria is nitrification, and the bacteria are nitrifying bacteria. And what is important about these bacteria, that these nitrifying bacteria are chemosynthetic bacteria. That means they are autotrophic. They can synthesize their own food with the help of chemical energy. They are not photosynthetic, they are chemosynthetic in nature. Okay, so first of all, ammonia is oxidized to form nitrite, and this is done by two bacteria, Nitrococcus nitrosomonas. Question directly comes. Then further, this nitrite is oxidized to nitrate, and this is done by Nitrocystis and Nitrobacter. Clear? So these four are the nitrifying bacteria which are chemosynthetic, autotrophic in nature, and they do nitrification. This slide is very important. Clear? Let's move on further. Ammonification. I just told you that when the dead matter is present in the soil, some bacteria decomposes that dead matter and releases ammonia into the soil. That is ammonification. And this is done by the bacteria Bacillus vulgaris or Bacillus ramosus. So this is mainly done by Bacillus species. And then comes denitrification. Denitrification is conversion of the oxides of nitrogen again to N2, and this is done by denitrifying bacteria. And the two important examples of denitrifying bacteria are Pseudomonas and Thiobacillus. Thiobacillus is again a chemosynthetic bacteria. Okay, so very important. These all are the NCERT examples. You have to learn them along with me. Pseudomonas, Thiobacillus. Okay, let's move further. Yes, these are the lines of NCERT. Let's see. The most important relationship is the legume bacteria relationship. You know what is legume? Leguminous plants. So a rod-shaped bacteria, Rhizobium, is found associated with several leguminous plants. Alfalfa, sweet clover, sweet pea, lentils, garden pea, broad bean, clover bean. These all are the leguminous plant. Any plant can come and they can ask which bacteria is found associated with this plant. So these are all the example of leguminous plants. Right. Then comes the example of Frankia here. Wow. Frankia. Frankia is found associated with a non-leguminous plant like Alnus. Okay. Let's move further. Yes. Now let's understand the mechanism behind the nitrogen fixation, biological nitrogen fixation. The mechanism behind the biological nitrogen fixation. So, to understand this, let's understand this diagram given in the NCERT. So, this is the root of the plant, right? Suppose I have taken a leguminous plant. This is a leguminous plant, right? It's a short plant. It has just germinated, right, from the soil. It's a small plant, and these are the roots. So, what it will do, it will release some chemicals. it releases some chemicals like betanes. The root of the plant, it releases chemicals into the soil, and those chemicals, they attract the rhizobium bacteria present in the soil. So the rhizobium bacteria which is present in the soil is attracted towards the root, right? So first step is the plant release chemicals into the soil, and because of those chemicals, the rhizobium gets attracted towards the root of the plant. So here the bacteria, so you can see in the diagram given in NCERT that the bacteria is getting attracted to the root hair and it gets accumulated here. Then second step, this bacteria is responsible for the formation of hook, and this is called as curling of root. This is called as curling of root. So second step is, this is the first step. The second step is curling of root, and this curling of root is done by nod factors released by bacteria. Right? Bacteria releases certain factors called as nod factors, and these nod factors, they curl the root, and this is called as curling of root, right? Then what happens? The bacteria forms an infection thread. So third step is formation of infection thread. Third step is formation of infection thread. Formation of infection thread, right? And with the help of this infection thread, the bacteria reaches to the cortical cells of the root, right? This I will teach you in plant anatomy, that when you cut a root, what you see outside, the outermost layer in a root is that of epidermis. Below epidermis, a root has cortex. Below cortex, a root has endodermis, and then pericycle, right? So these are different layers in a root: epidermis, cortex, endodermis, pericycle. Clear? So what happens that with the help of infection thread, the bacteria reaches the cortex and the pericycle cells. And what does it do? You know that parenchyma is present in the cortex and pericycle, and parenchyma is not the dividing tissue, right? So what happens over there? Let's see. So fourth step is, so beta, what is happening in the fourth step? Bacteria reaches the cortex and pericycle, right? It reaches the cortex and pericycle of root, and what it does, what happens over there? There is proliferation of cortical cells. There is proliferation of cortical and pericycle cells, due to which nodule is formed. What I'm trying to say over here is, let me tell you one thing also. Because of auxin by plant and cytokinin by bacteria. So understand what I'm trying to say here. The bacteria, with the help of infection thread, has reached to the cortex and pericycle. And what the bacteria is doing, it is giving plant the signal, I have come here, I am arrived. So when the plant receives the signal, plant forms large amount of auxin and the bacteria forms cytokinin. And auxin and cytokinin together, what they do, they say, they give command to cortical cells and the pericycle cells, divide uncontrollably and form a tumor-like structure called as nodule. This is called as proliferation of cortex cells and pericycle cells, uncontrolled division, rapid division. And because of the rapid and uncontrolled division of cortical and pericycle cells, a tumor-like structure is formed, and that is called as nodule. Clear? So this is how a nodule is formed inside the leguminous plants, the root nodules. Clear? Now, the question is, what happens inside the root nodule? So this is how a mature nodule is formed. Right? Now let's see. And yes, one more important point, NCERT has written it. A mature nodule is complete with vascular tissues continuous with those of the root. That means this mature nodule, which is formed inside the root, is in contact with the xylem and phloem of the root. That means the bacteria inside the root nodule is deriving the nutrition from the plant. Right? That is why this is symbiotic association. So this mature nodule is in direct contact with the vascular tissue of roots. Okay? Yeah, let's move forward now. Now let's see what's happening inside the nodule. Inside the nodule, now nodule has been formed, right? This is nodule. This is the nodule. Right? And I have zoomed this nodule to make you clear that what is present inside the nodule. So you can see here that there are group of non-dividing bacteria. Right? This group of non-dividing bacteria present inside, because this bacteria will not divide anymore, because this has to perform a function, and that function is nitrogen fixation. So this group of non-dividing bacteria inside the nodule is called as bacteroid. Bacteroid. Okay, and NCERT says that they are rod shaped. Clear, beta? So nodule is formed now. And inside the nodule, the bacteria is present, and the bacteria has to perform some function now, that is nitrogen fixation. That is why it has become non-dividing in nature. Now, the bacteria sitting inside the nodule wants to fix the nitrogen. But it is saying that I have a very strong enmity with oxygen. If there is any oxygen inside the nodule, I will not fix the nitrogen. Okay. So the plant is saying, okay, I will take care of it. I will make a compound which will clear all the oxygen from the nodule. It will take care that no oxygen is left inside the nodule. So a special compound is prepared here, and the name of that is leg hemoglobin. Leg stands for leguminous hemoglobin. The heme part in the hemoglobin is formed by bacteria, and the globin part is formed by the plant. That means the leg hemoglobin is formed by the participation of both bacteria and the plant. So this leg hemoglobin is a pink colored compound which is formed inside the nodules by participation of both bacteria and plant. And what is the function of leg hemoglobin? To clear all the oxygen from the nodule, right? So it binds with all the oxygen present in the nodule, and that is why leg hemoglobin is also called as oxygen scavenger. It is also called as oxygen scavenger because it clears all the oxygen from the nodule. Okay, so now what will happen next? Now the bacteria will synthesize an enzyme. The bacteria will synthesize an enzyme called as nitrogenase enzyme in the root nodule, and this nitrogenase enzyme can properly function only in the absence of oxygen. This has a strong enmity with oxygen, right? What are the special points regarding these enzymes? It's a molybdenum iron protein. It is a molybdenum iron protein. That means iron and molybdenum are a part of this enzyme. Very important question. Second, it works only in anaerobic conditions, right? And this enzyme is only present in nitrogen fixers. On Earth, this enzyme is only present in some bacteria which are nitrogen fixers. And it is coded by the NIF gene. The NIF gene present on bacterial DNA codes for this particular enzyme. And what is the function of this enzyme? To fix N2 to form ammonia, right? Now there is a very important equation given in NCERT, a balanced equation of ammonia formation. One molecule of nitrogen combines with 3 molecules of hydrogen in the presence of 8 electrons, 8 protons and 16 ATP. High amount of energy is required to break N triple bond N. Then in the presence of nitrogenase enzyme, 2 molecules of ammonia, 1 molecule of hydrogen, 16 ADP, 16 phosphate are formed. This is a balanced equation of nitrogen fixation given in NCERT. Now, how the question can confuse you? If the question asks how many ATPs are required to fix one nitrogen, what will be your answer? How many ATPs are required to fix one nitrogen? You will say 16. If the question says how many ATPs are required to form one ammonia, the answer will be 8, right? When one nitrogen is fixed, 2 ammonia are formed. So to form one ammonia, 8 ATP. To form 2 ammonia, 16 ATP. To fix one nitrogen, 16 ATP. Clear? So learn this equation. This is an important equation, balanced equation given in NCERT. So this is nitrogenase enzyme, right? This is the nitrogenase enzyme. N triple bond N is getting binded up with this enzyme. Then 2 hydrogen atoms are coming. Then it is formed. Here it is N2H2. Then again 2 hydrogen comes. It becomes N2H4. Then again 2 hydrogen atom comes. Then it becomes 2 molecules of ammonia. So this is how this enzyme catalyzes the formation of 2 molecules of ammonia. Okay. Now let's move forward. Now we have understood that how the nitrogen gets fixed up by nitrogen fixers. Now let's understand how the plants uses up this nitrogen inside them. This is called as assimilation of nitrogen inside plants. Let's understand this. Yeah. So beta, this is very important that the plants have taken up ammonia from the soil. Okay. But, and this ammonia can be taken up in the form of ammonium ions, okay? Because ammonia can never be like in the form of ammonia. It forms ammonium ions by combining with water. So, plant has taken the nitrogen from the soil in the form of ammonium ions. Okay. But the problem is that ammonia is toxic to the plants. So, plant cannot allow this ammonia to be accumulated inside them. So they will have to find a way to use this ammonia, right? Because this ammonia cannot be accumulated in the plant. So plant has found out a way to use this ammonia instantly. And the name of that way is reductive amination. How the plants use this ammonia? They do reductive amination. Now let's see what is this reductive amination. In this process, we take a keto acid. We take a ketone compound, ketone group containing acid called as keto acid. And alpha ketoglutaric acid is a 5-carbon keto acid. So the plant takes the help of a keto acid. It says, please accept these ammonium ions. We have to consume these ammonium ions because we cannot allow them to accumulate. So alpha ketoglutaric acid says, okay, I will accept these ammonium ions. So this keto acid accept these ammonium ions, and we need a reducing agent here also, and the reducing agent is NADPH. So what we are actually doing with keto acid? What actually we are doing with keto acid? We are reducing the keto acid with the help of a reducing agent, and we are adding ammonia to it. So we are aminating. Amination means, what is the meaning of amination? To add amine group, to add ammonia to any compound. So reductive amination means, what is reductive amination? That we are reducing a keto acid and we are aminating a keto acid. So reductive amination of keto acid is going on here. Reductive means reducing, and amination means addition of ammonium groups, ammonium ions, right? So as soon as we add ammonium ions to the keto acid in the presence of reducing agent, then this keto acid get converted into amino acid. That is glutamic acid. And the enzyme used here is glutamate dehydrogenase. So how, see, plant is so clever. How plant has consumed these ammonium ions? The plant has formed amino acid by these ammonium ions. So when a keto acid accepts ammonium ions in the presence of a reducing agent, it gets converted into amino acid, and this is reductive amination. And when alpha ketoglutaric acid undergoes reductive amination, the amino acid which is formed is glutamic acid, or you can also call it as glutamate. And this is the primary amino acids of plant. That means with the help of this amino acid, we will form different types of amino acid. How? I will tell you. Okay. So this is reductive amination, beta. Clear? The enzyme used up here is glutamate dehydrogenase. Now, another process, transamination. The term is by itself defining it. Trans means transfer. Amination means amine group. In this process, you are transferring amine group from one molecule to another. So what's that? This is a keto acid and this is an amino acid, right? You know what is the basic structure of amino acid. carbon, alkyl group, carboxylic group, and NH2 group, right? This is the basic structure of an amino acid. So a process in which an amino acid transfer its amine group to a keto acid so that this keto acid gets converted into amino acid. You know, as soon as a keto acid accepts NH2 group, amine group, it becomes amino acid. So this amino acid here, has donated its NH2 group, to the keto acid. And as soon as this keto acid accepted this NH2 group from amino acid, it has been converted into other type of amino acid. And this amino acid, as soon as it has donated its NH2 group, it will get converted to the keto acid from which it is formed. Clear? From which it is formed. So what we can say that during this phenomenon, during this process, we can form many type of amino acids with the help of one amino acid. Right? How? I will just tell you. And the kind of enzymes which are used in this process are aminotransferases or transaminases. Amino matlab enzymes which are responsible for the transfer of amino group, or transaminases, enzymes which are responsible for transfer of amine group. So two names of same enzyme, aminotransferases or transaminases. Okay? Yes. So this is the diagram of NCERT. Amino donor. Who will be the amino donor? The amino acid, right? The amino acid. So here I am taking the amino donor as the amino acid, and that primary amino acid is glutamic acid. Right? I have just told you that glutamic acid is the primary amino acid of plant, because with the help of this amino acid, I will make different kinds of amino acid in the plant. This is the keto acid, which will accept the amine group. And this is the oxaloacetic acid. Which is a 4-carbon keto acid. So this amino acid will donate its NH2 group to this keto acid, will transfer its NH2 group to the keto acid, right? And as soon as this amino donor will donate, so it will get converted, this amino donor will get converted into the keto acid, right? From which it was formed. And glutamate was formed from which keto acid? Alpha ketoglutaric acid. And this keto acid, as soon as it accepts NH2 group from the amino acid, it will get converted into another type of amino acid. And this another type of amino acid is aspartic acid. Or aspartate. This is transamination. And the enzyme used here is glutamate aspartate transaminase. Glutamate aspartate transaminase or aminotransferase. Are you getting my point? So here, with the help of glutamic acid, I have made another type of amino acid, that is aspartic acid. So this is transamination. Now, the plant has made different kinds of amino acids. Now the plant wants to make amides now. Now what are amides? Let me tell you. This is the basic structure of amino acid, right? A single amino acid has one NH2 group and one carboxyl group, right? So what happens when an amino acid gets converted into amide? This carboxyl group, COOH group, the OH part of this carboxyl group is replaced by NH2 group. And as soon as this is done, this gets converted to this. See, carbon, 1 NH2 group, CO. In place of OH, what has come over here? NH2. So CONH2. So this is amide, right? So what's the difference between amino acid and amide? The carboxyl group, if the OH group is replaced by NH2, it will become amide. So what you can understand here, that a single molecule of amino acid contains only 1 nitrogen, but a single molecule of amide contains 2 nitrogen, right? Now tell me one thing. If a plant has to, translocate nitrogen from one place to another, in what form it will prefer to translocate? It will prefer to transfer nitrogen. Think. Obviously, if this nitrogen has to travel through the xylem and this nitrogen has to be transferred through xylem, the plant will prefer to transfer this nitrogen in the form of amides. Why? Because a single molecule of amide contains 2 nitrogen. When a single molecule of amide will be flowing through the xylem vessels, along with it 2 nitrogen atoms will go. Are you getting my point? So what I have told you up till now, that how plant use this nitrogen inside them. So they have taken ammonia, right? But the problem is this ammonia cannot be accumulated in the plant. So plant has found a way to use this ammonia by combining these ammonium ions with the keto acid, and they form amino acid. And the primary amino acid of plant is glutamate. With the help of glutamate, plant forms other different types of amino acids by the process of transamination, where the glutamic acid donates its NH2 group to other type of keto acid, and that other type of keto acid gets converted into other type of amino acid like aspartate. So how it has used that ammonia? First, it has converted it to amino acid, then it has used that amino acid to form different kinds of amino acid, then it has formed amides from amino acids. It has formed amides from amino acids. See, this is how finally the plants use this nitrogen. And the plant prefers to translocate nitrogen through xylem vessels in the form of amides, right? And why amides? Because a single molecule of amide contains 2 nitrogen. And now the last lines of NCERT, that in cases of leguminous plants, I have just told you, in the leguminous plants, where is the ammonia getting fixed? Inside the nodules. So inside the nodules, finally the ammonia gets converted into urides. And if the nitrogen has to be exported from the nodules, right? Because in the leguminous plants, most of the nitrogen is present inside the nodules. So if this nitrogen has to be exported from the nodules to the different parts of the plant, that is done with the help of urides. Urides are nitrogen containing compounds. So see what is written over here, that in plants like soybean, the nitrogen is exported in the form of urides. So you can say that in leguminous plants, in legumes, nitrogen is exported out from the nodules in the form of urides. Why? Because these urides have a large amount of nitrogen in them. See what NCERT is saying? These compounds have high nitrogen to carbon ratio. They have high important. If the option says that urides have high carbon to nitrogen ratio, that would be false. Urides have high nitrogen to carbon ratio. That means they have high amount of nitrogen. That is why the leguminous plants prefer to export the nitrogen from nodules to different parts of the plant in the form of urides. Okay? So, the chapter is finished. I hope you understood the chapter well and you have learned the things along with me. So, beta, if you don't understand anything, just pause it, rewind it, listen it again, and learn the lecture learn the things along with the lecture. And after that, you can read your NCERT line by line. I have covered almost all the NCERT here, and your chapter will be prepared. Okay? So, thank you and all the best.